Hello, my name is Gene Williams, Executive Chairman of ProMIS Neurosciences. Thank you for your interest in ProMIS. The topic I'll be covering today is the opportunities that brought me into ProMIS Neurosciences when we did a strategic reorganization last summer, which is the opportunity to develop a portfolio of precision therapies for Alzheimer's disease. I'll be making some forward-looking statements. Start us with a summary of the story. We're developing a portfolio of disease-modifying therapies targeting the prion. To be disease-modifying, to fundamentally change the course of disease, you have to address the cause of disease. What we know now is that it's the prion form of beta amyloid. I'll talk a little bit more about the science later, but there are multiple forms of beta amyloid: monomer, plaque, prions. The prions are the bad actor that kill neurons in your brain and drive disease. So we'll be focusing on precision therapies targeting prions. There are multiple strains of prions, so we'll have a portfolio of therapies. Well, why do we know that's the case? In part, we know based on an analysis of a lot of the large pharma programs that have come before us and the amazing amount of spending and investment that's been made in the area of beta amyloid. Beta amyloid is by far the most invested in, the most studied, and therefore the best understood currently area of promising treatments for Alzheimer's disease. We're not focusing on some novel area of biology which has been untested. What we're trying to do is take our proprietary skills and technology, take an area which has been tremendously well understood based on tons of investment over many years, and drive it over the goal line. The way we do that is by creating prion selective therapies. What I'll do is I'll show you some data from various different large pharma programs that have pointed us the way to where the goal line is. ProMIS's our opportunity, ProMIS's objective is to drive us over the goal line to having safe, effective therapies that modify Alzheimer's disease. Well, how do we think we can do that? We have competitive advantages based on our scientific team. Our scientific team of Chief Science Officer Neil Cashman and Chief Physics Officer Steve Plotkin have spent their entire careers figuring out how you do precisely this, how you identify therapies that selectively target a prion and leave alone the other forms of a protein like beta amyloid. We have patented processes, we have decades of experience, we have the capability to do in months for tens of thousands of dollars what might take larger companies years and tens of millions of dollars. And so far, that capability is proven out, as shown by the various patent filings we've announced over the last few months. As you know, and probably why you're listening, is that Alzheimer's is one of the biggest opportunities left in medicine. We'll talk about some of the patient numbers in a moment, but the market value numbers are also pretty incredible. If you look at examples of companies that have phase 2 assets in Alzheimer's disease, some not even disease modifying, the market values tend to be in the billions of dollars. Axovant went public with a product that helps increase the value of therapies on the market by extending cognition for a few months. Valuable thing, very valuable thing. Market cap between $1 and $3 billion. There was recently a deal announced with Heptares for a total of $3.3 billion. Also, phase 2 assets, similar type of capabilities. Biogen, when it announced the first ever successful but not completely successful because of side effects, results for their product aducanumab, a beta amyloid modifier, their market cap went up by $20 billion. So there's tremendous market value potential. We plan as a company to take our therapies through phase 2 and try to get to the same level as those companies I've mentioned. We think the opportunity will continue to be significant. And that could lead us to an M&A exit in 3 to 5 years. Now let me drill a little bit deeper into the science. Let me show you a picture of Alzheimer's quite literally. What you see on the left is a healthy, normal brain that has 100 billion healthy neurons with synaptic connections. That's what helps all of us remember our friends and remember where we are. If you look at the brain on the right, that's an Alzheimer's brain. Look at all that black space. Look at all that brain atrophy. Billions of neurons have been killed by prions over a period of decades, getting to that level of Alzheimer's disease. The picture in the middle is mild cognitive impairment. That's when you first have symptoms. Look at how much black space there is then. So it tells us that the brain has been suffering from prions killing neurons for many years before we start to see symptoms. And our view is that if you can interrupt that process at any stage, the brain's amazing resiliency and plasticity could allow it to at least stop progression and possibly even restore some function if we can interrupt the disease with some fundamental disease-modifying therapy like the ones we're trying to develop. Give you a sense of some of the magnitude of the numbers. There are 5 million patients in the U.S. that have Alzheimer's. This is all information from the Alzheimer's Association. 5 million with Alzheimer's disease, 15 million with mild cognitive impairment, and a $500 billion total cost. That's both direct and indirect medical costs. Those numbers could double or triple over the next few years if we don't come up with the types of therapies that ProMIS is working on right now. Would bankrupt Medicare if we're not able to do something about it. Fortunately, there's light at the end of the tunnel, and I think that we will have some very, very good therapies from ProMIS and from others, over the next few years to make a difference. Now, let me take you a little bit into that science that I talked about at the beginning, the prion. The way beta amyloid comes about in your brain is a protein called amyloid precursor protein, which is indicated, which is helpful in the synaptic formation. When it breaks down, as all proteins do, it secretes beta amyloid monomer, which is a single fibril, as a single strand, as you see there. Beta amyloid monomer wants to aggregate, so it aggregates into plaque. In fact, in the very, very early days, the reason why beta amyloid was targeted was because scientists saw amyloid plaque in the brains of patients who had died of Alzheimer's disease. And, we had the analogy of cholesterol lowering and plaque in your arteries, and that was a very, very telling analogy. But the problem was it was only half right. People started focusing generally on beta amyloid, and particularly on the plaque. What we've come to learn, though, after many, many decades of that not getting all the way to the success that we wanted, is in fact there's a different process going on. In fact, this was a point of view held by our chief scientist, Neil Cashman, and some of his colleagues who had been focusing on prion science for decades. This is actually what goes on that drives the process of killing neurons in your brain. When beta amyloid monomer is produced, sometimes it aggregates into a neurotoxic prion. A few monomers get together, 5, 6, 7 of them, something in that range, and they take on a misfolded shape that is neurotoxic and propagates. The way it propagates is other beta amyloid monomer, when it's in the vicinity of a prion, also misfolds in a prion-like shape, which is how it spreads, and what leads to the neuronal death and the cognitive decline. Now, since large pharma focused on the top view of the world, a lot of the therapies that they developed didn't actually have the benefit that we were looking for. That's why we're shifting our focus at ProMIS Neurosciences. This is a fairly recent consensus that prions drive neurodegenerative disease. Again, prions are the misfolded neurotoxic form of proteins that might otherwise be healthy or inert that propagate and drive disease. At the Academy of Neurology meeting, September 2015, the state of the consensus was clearest. The president's plenary session was about the fact that we all agree now that prions drive neurodegenerative disease. After years of vigorous debate and differences of opinion, the data were pointing in that direction, and that was the strong consensus. Our CSO, Neil Cashman, is one of the featured speakers at that meeting. Another aspect of this finding, however, this consensus, this emerging consensus that prions drive disease, that have implications for what you need to do for effective drug development. This is something that our science team has a tremendous amount of experience with. These implications are very, very important. Prion variants are a small percentage of the total protein population, so there's a lot more beta amyloid monomer, a lot more plaque in the brain than there are prions. What that means is if you don't have therapies that very, very selectively target the prion, you'll either waste your ammunition binding to the monomer, or you may cause side effects binding to the plaque. And that's, in fact, exactly what we've seen in some of the big pharma programs that I'll show you in just a moment. Now, the other important part that I won't go into in too much detail today, but it's part of the reason why we're developing multiple programs against 6 different targets, is that there are different prion strains. We believe there are going to need to be 3 or 4 or 5 complementary therapies that deal with different strains of prions. Different patients have different strains. You need to target the right strain with your therapy in order to give that patient the disease-modifying therapy that they need. Now, let me show you these big pharma programs that unintentionally, pave the way towards understanding where the goal line is. We're leveraging all of this information. I think each of these big pharma programs, to the best of our knowledge, was trying to target beta amyloid broadly and come up with a monoclonal antibody, which binds very, very effectively to beta amyloid. And they ended up binding to different forms of beta amyloid in a way that really illustrates where the goal line is. Bapineuzumab was the J&J Pfizer product that bound to both monomer, bound to plaque. So by binding the monomer, it had no efficacy. By binding to plaque, it had side effects. It didn't work. Solanezumab, on the other hand, bound to monomer, so it missed its primary efficacy endpoint, but it didn't bind to plaque, so it didn't have those side effects. The side effect I'm talking about is called area E, amyloid-related imaging with edema. It's a vascular edema, an inflammation in the brain that really is serious, so it forces you to drop the dose. So solanezumab now has been studied in ongoing studies, and because it didn't have the side effect, because it didn't bind the plaque, it may find a role in earlier stage patients or at a higher dose. Biogen's aducanumab was the first product to really re-energize the beta amyloid area after a long track record of failures and disappointments. Biogen's aducanumab showed efficacy for the first time on a scale that's 30 points, the MMSE scale. Patients in the high dose arm didn't progress at all, essentially, and patients in the placebo arm progressed by 3 points. A very meaningful clinical difference. The reason it had efficacy, we believe, is because it didn't bind the monomer. However, because it bound plaque, it had that side effect, area E, and had all the patients in the high dose arm had to back off. So we believe what that tells us that the winning product will be prion selective, not binding the monomer, not binding the plaque. Hundreds of millions of dollars of prior spending by large pharma tell us that's the target product profile that looks like it'll be a significant winner. And that's exactly what we're trying to develop. We're underway developing that right now. Precision portfolio, binding to the correct epitopes. Now, the epitope is the portion of a protein, like beta amyloid, that a monoclonal antibody recognizes. Think of it as the targeting location. You need to get that very precisely correct in order to target just the prion and leave alone the monomer and the plaque. And as I mentioned before, that's what our science team has spent their entire career doing. And over the last few months, they've proven to be extremely efficient and effective at doing that. Where we are now is we're pretty well underway in developing those products. Let me show you our status. We've run our computational methods and applied our scientific experience. Our science team has come up with 6 novel targets, which could account for different strains of prions, all of which we believe will be selective for the prion, avoid the monomer and the plaque. But we want to make sure that we validate that. Well, we know we want to have happen in the brain of a patient with Alzheimer's disease. We want our products to bind the prion, but not bind the monomer or the plaque. How better to show that than by using brain tissue from patients who died of Alzheimer's disease and donated their brains to. And that's exactly what we're doing. It's called in vitro validation. So we'll have multiple assays that show that we bind prion, don't bind monomer, don't bind plaque, and have some functional benefit in stopping propagation and stopping neurotoxicity. That'll lead us to some number of validated products, validated epitope targets, which are ready to be developed into fully human therapeutic products. That next stage is to create both monoclonal antibody therapies, which we do by humanizing those original mouse products that we use for validation, and then creating a companion diagnostic. What that means is a diagnostic where you could take some spinal fluid from someone who may have Alzheimer's disease, test that spinal fluid and say, we detect this prion, we detect a different prion, and know exactly which therapy is necessary to treat that patient's specific prion, the one that's killing neurons in their brain. We're applying a few approaches to mitigating risk. As I mentioned before, a lot of our choice of target, our choice of focus as a company was identifying an area where our proprietary science would enable us to drive a knowledge base that had been accumulated over many years after many hundreds of millions of dollars over the goal line, which is an understanding about the prion selectivity that you need in beta amyloid. In addition, we're taking multiple shots on goal. Also, we're using monoclonal antibodies as therapy for a number of reasons. One, this has been the area where most of the previous therapies, which has been the type of therapeutic approach most of the previous therapies have used. So there's no reason a priori to believe that our slightly differently targeted monoclonal antibodies against beta amyloid will have different safety issues or manufacturing issues. In fact, the odds are very, very good that those will work out very favorably. So we're mitigating risk by going down those paths that have been trodden pretty well before. And by using companion diagnostics, we're going to be able to make sure that in our clinical trials, all the subjects who are involved or who are enrolled actually have the prion that we're blocking. In many previous trials, patients were enrolled who didn't have Alzheimer's disease or didn't have the form of Alzheimer's disease that might be addressed by the therapy that was being used. We're going to reduce that risk to a minimum by having companion diagnostics. So when you put together all of these factors, we think we're choosing a path that dramatically mitigates risk versus many other programs that are going on in Alzheimer's. Let me end by showing you sort of a 5-year plan for building value to a possible M&A exit. First, with our primary therapies, up to 6 precision therapies in Alzheimer's, it'll be about a 2-year process once we have those validated between the middle and the end of this year to do manufacturing scale-up in animal toxicology or animal safety, after which we'll be filing an IND. In parallel with that, what we'll be doing to help understand better the role of different prions in disease is we'll be creating a companion diagnostic for all of the different therapies that we're developing, and then doing an Alzheimer's cohort study. We'll take spinal fluid from many patients with Alzheimer's, and we'll be able to understand the prevalence of different prion strains, or which prion strains might be associated with the most rapidly progressive disease. Those would be two important features for us to be taking into account when we think about prioritizing our products going forward. We already have 3 products in the ALS area. We're actively seeking a partnership for those products. They're also prion selective against a different protein, SOD1, which drives ALS the way beta amyloid drives Alzheimer's. We're seeking partnerships there so we can focus all our resources and energies on the opportunity we see for prion selective beta amyloid precision therapies. When we get into the clinic, we'll have to do a phase 1 safety study first, and then we'll do 2 clinical trials, one in Down syndrome and one in Alzheimer's. The one in Alzheimer's we think will look very much like the Biogen aducanumab study, with the difference that we'll be selecting patients with a companion diagnostic, so we'll know that every patient in our trial has the prion that we're blocking. And it'll also be early stage patients and hopefully a roughly similar size. The reason why we're picking Down syndrome as well is because you may have seen in the Wall Street Journal a couple of months ago that Down syndrome patients have a chromosomal mutation that causes them to make excess beta amyloid through monomer because they have more amyloid precursor protein than most of us have. Because there's a lot more monomer around, more opportunities to misfold into prions, more monomer to recruit into other prions once those start to gain critical mass and grow. The net result is Down syndrome patients have Alzheimer's disease in addition to any mental retardation that could be part of the disease process. They have Alzheimer's disease driven by beta amyloid as early as their teens, and by age 65, 75% have Alzheimer's disease. So we think that could be a very, very important opportunity for us or the partner with who eventually takes these programs through approval. Down syndrome could be an orphan designated disease, and it could be a very rapid and cost-effective means to get to an initial approval. I think that could lead us to a possible M&A exit in a 3 to 5 year time frame. So wrapping up again with the investment thesis, developing a portfolio of disease-modifying therapies because they target the prion. There are different strains of prions, so we think 3 to 5 therapies will be necessary to cover the population. We have consciously chosen an area where prior investment, more investment than any other area in Alzheimer's, has gone into beta amyloid. We think that gives us the opportunity to take a path where we're driving things across the finish line based on standing on the shoulders of giants who have come before us and make significant investments. We've applied, the competitive advantage that our team has developed over decades of their scientific career. We have proprietary processes, some of them covered by patents, that allows us to identify the key to prion selective therapies, which is the epitopes that are uniquely associated with prions that aren't recognized on the monomer and on the plaque. And we think that could lead us to an M&A exit in 3 to 5 years. Thanks very much for your time and attention, and please contact us at with the contact information seen here if you'd like to learn any more. I appreciate your time.